Okay, thank you. So, good afternoon and good morning to our Canadian friends. We are now on the second day of this winter school organized by Coris and the University of Padua and Genome Quebec. Today, the table is devoted to gene therapies for rare diseases. And so there will be top scientists discussing this topic. And Alessandra will be the moderator who will lead the discussion following the presentation. Now it's my pleasure to introduce Manon Pedetier. And Manon is from the Fonds de Recherche Santé Quebec. Correct me if I'm wrong, Manon. And so,
wrong.
Manon, if you would like to add something.
So hello everybody, bonjour, good morning, buongiorno. So I'm Manon Pelletier. I'm glad to represent the Fonds de Recherche Québec Santé, as well as our partner, Genome Quebec, to this Coris Winter School for Gene Therapy for Rare Diseases. So just to let you know that the Fonds de Recherche Québec Santé is the Quebec funding agency for health research. Its mission is to provide financial support for all areas of research in the field of health, to support the training of graduate students and careers of researchers, as well as financing the research centers and thematic networks. Genome Quebec is a not-for-profit organization with a mission to catalyze the development and excellence of genomics research and accelerate the discovery of new applications for genomics in health. It's really a pleasure to be here. So wish you great discussion on this great topic. Thank you.
Thank you, Manon. So just one piece of information that Professor Yannick Doyon will not be present at this table today. So we are going to close a little bit in advance. So there will not be a break between presentation and discussion. And this is an information for Alessandra Bifi, who will act as a moderator. So please, Alessandra, take the lead.
Thank you, Gino, and thank you all for this initiative. It's really a pleasure for me to discuss with you today about gene therapies for rare diseases that we will address on from a broader perspective, beginning from the diagnosis and ending up into the efforts we are willing to accomplish in order to provide to every patient with a genetic diagnosis and opportunity for advanced treatment, possibly with gene therapies. Just a short introduction about myself, and I have been asked by Gino to lead the discussion today. I arrived in Padua 3 years ago as professor of pediatrics and chief of the Pediatric Hem-Onc and Stem Cell Transplant Unit here in Padua. And previously, I was working in Boston as director of the gene therapy program at Boston Children's Hospital. And I would say that gene therapy has constituted the roadmap of my career and the basis of my discoveries and clinical developments. And in particular, during my career, I've been really working on the opportunity from moving from the bench to the bedside and back for rare diseases, genetic disorders, and I think this would be the theme that we will be discussing about today. So the session is planned in two areas. We have a first area where the speakers contributing to the discussion will introduce their research and give us the basis for discussing about potential areas of shared interest, and then a second part where we'll be addressing a few questions I listed and I share with the discussants, really focusing on advanced diagnostics, novel therapies, and what we can do to really move forward in implementing new treatment opportunities based on this. So the participants to the discussion today are Cristina Basso, Professor of Pathology at the University of Padua, Alexei Tseltresky, Professor at the Department of Pediatrics in Montreal, Maurizio Muraca, who's Professor of Internal Medicine Medicine here in Padua, Fabien Touzou, Clinical Associate Professor at the Department of Pediatrics in Montreal, and Leonardo Saldiatti, who's Professor of Genetics here in Padua. I would invite all of them to introduce themselves and their research, starting from Professor Basso.
Okay, good morning to everybody. First of all, let me thank Gino Gerosa for the invitation to this winter school. Winter school are very important meeting for researcher because invitation to exchange internationally experience in research in course in science. And by the way, I'm appointed recently as a Vice Director for International Relations, so I need to be fully supportive of this kind of initiative because it's the only way to go when we are dealing with rare disease like the topic of today. As far as my background, I've been already introduced. I'm a pathologist, but basically I'm not a general pathologist. I started as cardiologist, then I decided to move to pathology. So my topic is cardiovascular disease since the beginning, with a special target of clinical pathological correlation, and among my interests, scientific interests, are inherited cardiomyopathies at risk of sudden death. Probably this is why I'm here to discuss with you a little bit what we are now in the research in these inherited rare disease which are at risk of ventricular arrhythmias and sudden death, particularly in young people. That for now is enough.
Thank you, Cristina. Alexei, introduce briefly yourself.
Right. Well, I actually prepared a small PowerPoint, so which I would like to share now with you. I hope it wouldn't be too long. So yeah, it's great to be here. Thank you so much for invitation. And I don't know. Can you see my?
Yes. Yes.
In person, because winter school should be about skiing and sharing and talking. But I think it's coming because I just came back from the first meeting in presence, which was ward meeting on lysosomal disorders. So this is my 11th session, and I will be speaking about the therapy approaches we use to treat San-Felipe syndrome. And San-Felipe syndrome is belongs to the group of lysosomal storage disorders. It's about 50 rare genetic metabolic conditions that are caused by deficiencies of lysosomal enzymes, or enzyme or proteins involved in targeting of metabolites to and out of lysosomes, or proteins that are involved in the lysosomal biogenesis. And these are rare disorders, of course, but as a group, they affect about 1 to 7,000 of live births. So they are important conditions. And of course, some disorders are more frequent in specific groups of populations. And now, one subgroup of lysosomal disorders, mucopolysaccharidosis, or MPS, neurological MPS, is still remains untreatable and presents the most important health risk for the patients. And these disorders, MPS 1, 2, 4 subtypes of 3, then 7, are caused by the deficiencies of enzymes involved in the breakdown of glucosamine-glycans, heparan sulfate, dermatan sulfate, and keratan sulfate. And San-Felipe, or MPS 3, it's the disorder that manifests mostly neurologically. It starts with at 3-5 years with sleep and behavior disorders, and then results in cognitive defect and hearing loss. And then, of course, the dementia kicks in, and then it all results in the loss of communication, and finally, children die generally before adulthood. And we're interested in the subtype 3, which is caused by the defects in the transmembrane acetylation of heparan sulfate. So acetyl group is taken from acetyl-CoA from cytoplasm and transported by this enzyme, which is also a transporter, 11-domain transmembrane protein, and it's used to target heparan sulfate. So as for other lysosomal disorders, you can either target the primary genetic defect by gene therapy or stem cell therapy, or you can try to replace by reduce the production of the substrate using enzyme replacement therapy, chaperone therapy, or substrate reduction therapy. Then you can look at the different secondary defects and try to either induce autophagy or treat secondary pathological changes. And when we made the mouse model of this disorder, we found that actually for the brain, for the CNS disorder, the dysfunction of macrophages, of microglia cells in the brain, are the most important. So these cells store most of heparan sulfate in the lysosomes, and then they become activated. They secrete toxic cytokines, TNF alpha, MIP1 alpha, and this actually results in secondary changes in neurons. It's mitochondrial defects, accumulation of misfolded proteins, and so on. And this is fully recapitulated in the brains of patients with different forms of neurological MPS. You also see the accumulation of activated microglia and astrocytes. So basically, we followed the very seminal paper of Alessandra in Science, and we decided to try to use hematopoietic stem cell gene therapy. And this is what we do. We take from our mice, we take the bone marrow, we purify by negative selections hematopoietic stem cells, heat them with the lentivirus to induce about 10-fold overexpression of missing enzyme, HGS-NOT. And then we condition mice with busulfane and inject back, transplant them with this cells, and then look at the engraftment and then finally at 6 months, when normally we see pathology in these mice, we study their behavior and sacrifice. So this is the results of the preliminary experiments where we transplanted 3 mice, and we could reverse or prevent, I should say, such trait as hyperactivity, because these mice, as San Felipe children, they're hyperactive. So we could reduce the amount of distance they cover in the open field test, and also increase the duration of immobility. And then, unfortunately, we don't have very good engraftment in all these mice, but 2 were engrafted 82% and 1 only at 17%. And that actually reflected, we could see increased activity in bone marrow and spleen, but not in the brain. But nevertheless, we could see reduced neuroinflammation, like CD68 and LAM in the brain, and also reduced lysosomal signs and dependence. So basically that's it, and now we're concentrated on improvement on our vector, because this is key to achieve higher levels of the enzyme activity in transplanted mice. And also, you probably can ask how you cannot rely on cross-correction, right, of neurons from microglia by transmembrane enzymes, but we actually found that cells overexpressing HGS-NOT, they produce exosomes that are loaded with these transmembrane enzymes. And if we treat cultured cells with these exosomes, we can see uptake of the enzyme, which is GFP labeled, into the lysosomes of treated cells. and reduction of lysosomal storage. So maybe this will help us. And yeah, thank you very much for your attention. Right.
Thank you.
So let's hope. Yeah, I can answer questions now if you have any, or we will have like a discussion at the end.
Yeah, I think we complete the presentation of everybody, and then we can address the specific aspects of everyone's role, activities, and projects. I would
All
let Maurizio introduce himself.
Here, yes, I am online. Can you see my slide?
Yes, yes, we can.
Okay. Sorry.
Maybe Maurizio, you could keep this view, because in this way we might see all the slides. Otherwise, we would be stuck on the first one if you probably put them on full screen.
Well, there is.
If it's not working, I can do it for you, Professor Muraca.
Yes, please go ahead, because here it is completely blocked.
Okay, so please delete because I need to share for you. So you have to close yours, your presentation. I can do it for you.
Where do I close it? Here?
Um,
Okay, voila.
Okay.
That's it.
Can you see?
Yes.
Yes, I think we
very
can
good.
go to the next one. Okay. Next one, thank you. Well, my name then is Maurizio Muraca. I am an internist. I have been working for several years on extracellular vesicles. As you know, they are produced by practically every cell type, and they carry regulatory signals, and they can exert important metabolic effect on the target cells. Can you proceed, please, Georgia? We are studying in particular the use of these vesicles as therapeutic tools. Since they are small, they can easily cross barrier. They can show specific targeting. They carry effector and regulatory molecules, and even genetic information. Next slide, please. Since a few years, we have been working together in collaboration with the drug company in Belgium to develop clinical grade extracellular vesicles. And we have been performing, and hopefully we have concluded the preclinical evaluation of the mesenchymal stromal cell-derived extracellular vesicles for the treatment of bronchopulmonary dysplasia in extremely premature babies. We are currently developing what we call a second generation extracellular vesicles primed with the cytokines and exhibiting enhanced anti-inflammatory and anti-angiogenic activity. We are currently testing these vesicles both for the treatment of inflammatory bowel disease and of some ocular disorders. And finally, we are developing extracellular vesicles from engineered cells overexpressing SERPINB3 for oxidative stress protection. Next slide, please. Regarding rare disease, in collaboration with Professor Tetti from the University of L'Aquila, we have been studying extracellular vesicles from osteoblasts thanks to their specific targeting to the bone. We have been exploiting the specific targeting of these vesicles and their ability in particular to carry pro-osteoclastic functional activity, rank ligand specifically. Next slide, please. In this model of mice knockout for the rank ligand infused with the wild type extracellular vesicles from osteoblasts, we could demonstrate a dose-dependent increase of osteoclastic activity in the bone, as shown in these slides from the appearance of tartrate resistance acid phosphatase activity. We also showed that these vesicles are able to carry specifically to the bone therapeutic molecules like molecules that can be used for the treatment of different bone disorders. So that's all from my side. Thank you.
Thank you, Maurizio. Next, we have Professor Saldiatti.
Okay, thank you. Can you hear me? Because I just had some problems. I lost a part of Maurizio Muraca's presentation.
We can hear you very well.
Okay, perfect. Now let me share, because I have a couple of slides. And do you see the slides? Wait, yes. Okay, so my name is Leonardo Saldiatti. I am the director of the medical genetics program here in the University of Padova, and I didn't start out as a geneticist. I started out as a pediatrician, actually, and then I ended up in this field just by chance, actually. And our main research interests have been metabolic disease, although not lysosomal, but rather both mitochondrial and intermediate metabolism disorders. And among mitochondrial disorders, we have a special interest on coenzyme Q deficiency. As you know, coenzyme Q is one of the most crucial lipids within cells, because it plays several important roles in controlling cell life and cell death. The two main roles are ATP production within the mitochondrial respiratory and its role as an antioxidant. And coenzyme Q deficiency is one of the few disorders in which the classic paradigm of disease discovery was totally reversed. Instead, first we found therapy, then we found the biochemical diagnosis, and then at last we found the gene. In fact, we identified two patients who had this devastating renal and neurological disorder, and they actually responded to oral coenzyme Q supplementation, and this prompted us to investigate coenzyme Q deficiency and then to find the gene. What is interesting that these were two brothers. The first one was treated late in the course of the disease, after a few months after the symptoms had instituted,
when end-stage renal disease was already in place. So we did see improvement of the neurological condition, but no improvement of renal function. And whereas we had, since we had the information on her brother, we were able to treat his sister as soon as she started developing symptoms. And we were able to and preserve renal function for many years. The brother, in less than 4 months, he developed end-stage renal disease. The sister had normal renal for several years. So why we want to perform gene therapy for this disorder? Right? Because after 15 years of follow-up, even though the neurological picture is stable, the renal function is beginning to worsen. So so supplementation is actually worked, but it's not a long-term treatment. Certainly, we bought many years of normal renal function for this patient, but it wasn't. It is not the perfect treatment. And this is the same thing that happens in mice. And these are the mice, the untreated mice. These are the normal mice. These are the treated with coenzyme Q. And this line is what actually this has been published by the Gassi group. I corrected this figure. These are our data, so we know that even in the mice, long-term therapy is not perfect to preserve renal function. And so, what are the candidates for gene therapy? Certainly not the patients with a very severe neonatal form, for which very little treatment is available. The ideal patients are the ones who, with the milder intermediate forms, for which, and some of whom present only with isolated renal disease, as our model mice are using, that are these mice who are harboring spontaneous hypomorphic mutation in another gene involved in coenzyme Q, which display essentially the same phenotype as the patients do. And we are testing gene replacement using an adeno-associated virus, AAV9. And we chose this because there's very little experience with kidney, but there's a paper that came out last year in which Julie Saba, her group, showed that AAV9 was able to correct another glomerular genetic SGPL1. So we are now in the phase of we have built generated our vectors, and we are now testing the efficiency, the transduction efficiency in the kidney, and which is the most important, the critical issue, because apparently, even though there are some reports showing the efficacy of AAV9, kidney seems one of the most critical tissues to target with this. Now, what are the issues that we are dealing with? Well, the pros of our approach, we think, and I'd like to discuss this with you, and the fact that the gene we are targeting is only for glomerular cell function. So we just need to transduce the glomerular cells. We don't need to transduce the rest of the kidney. The rest of the kidney does not suffer. If you take out the same gene in the tubule, you have basically no effect. If you, whereas only if you target in the podocytes, you develop the disease. The other aspect is that probably very high efficiency is not required because there's evidence, and we are trying to demonstrate this. We have a model to demonstrate this. That once one cell is corrected, then there's physical transfer of coenzyme Q to the neighboring cells. So this is something that could help us succeed. The other point is that these genes are expressed at very low levels, so you don't need very high expression levels. And in this model, and in many patients, there's renal disease, so we have just to focus on the kidney. What are the problems? The issue is AAV9 the correct serotype to use for this, or are there any better serotypes? And are there any ways to improve delivery to the cells? And again, this is something that has been highly debated, although I'm here, I'd like to discuss this with you. And this is another point which is quite important, and because this protein, this PDS2 unit, is part of a multi-enzyme complex. And since the mutated protein is catalytically inactive, but it is quite stable, one of the problems that we could encounter if we have low expression levels, that there will be competition between the recombinant protein and the native non-functional but stable protein. So, and we tried to test this in cells, but we couldn't. In cells, we always got too much expression, so we don't know it in the animal. And the other point that we have to share, that we have to discuss, is whether there's a point of no return of renal damage. So, and this is critical for that tells us how critical is the early diagnosis for the disease. Finally, the whole point is to understand whether gene replacement is indeed superior to coenzyme Q supplementation, because a therapy that is at least partially is working and it's very cheap. This is going to be a quite expensive treatment, so it really must be demonstrate its great superiority to compared to the simple supplementation. So I'm done, and so again we can discuss later if Alessandra is okay.
Thank you, Leonardo. I would ask Fabienne if you could share with us some of yourself, your activities, your research focus.
Yes, of course. I'm sharing my screen. Okay. You see my screen?
Yeah.
Okay, fine. So thanks a lot for everybody. So a little bit of background. Me, I'm a clinical scientist. I'm a pediatrician, and I'm also immunologist. And I did my PhD on the subject of telomere biologic disorders and DNA repair in Paris. And from 2012 to 2016, I was responsible for the production of gene therapy products at the cell and gene therapy facility at the CHU Necker-Enfant-Balade in Paris. So I was involved in the GTP production for gene therapy trial involving SKIDX1 and also the HGB202 study, which was designed for the cure of patients with beta thalassemia and sickle cell disease. And I was fortunate to take part in the scale-up of a preclinical study for the treatment of chronic granulomatous disease, the X-linked form, and another project that was focused on the cure of patients with HIV infection and lymphoma. In 2016, I was recruited at Montreal to take part in in the life of the immunological department, and to develop a thematic of gene edition for modeling inborn error of immunity, and to develop also alternative strategy for the treatment of this disease. And at Saint-Just team, I'm the scientific director of the gene editing platform for the research center. So the gene editing platform of our center is mainly used right now to modelize some inborn error of inborn error of immunity, but also inborn error of other diseases for clinician or scientists that want to modelize some rare disease. And what we use as starting material is primary cells or cell lines or iPSC also, and we perform gene modification with CRISPR-Cas9 or Cas12 gene editing for knockout of genes or knock-in. We do also base editing, and recently we have developed a prime editing approach for modeling a disease, cell disease. My group has mainly focused right now on the modeling of autoinflammatory disorder because it's one of my main interests. So we generated different models for NOX2 deficiency, XEAP deficiency, RIPK1 gain of function, PSTP1 associated disease, mainly through CRISPR-Cas9 gene editing, but also with prime editing technology more recently. And what we did is to generate this model either on cell line for autoinflammatory disease, it's THP1 cell lines, monocytic macrophagic cell lines, but we more recently use iPSC cells that we are able to differentiate into macrophages. Another focus of my group is to try to improve the gene edition for primary cell in the aim of bring this therapeutic strategy for the clinic, because as you know, the rating of correction through homology directed repair is quite I would say not low, but not sufficient enough to allow the correction of gene defect for the majority of disease, at least in the field of immunology. So what we are trying to do right now is to try to enhance the homology directed repair through the introduction of modulatory proteins during the DNA break and the DNA repair. So we've got different candidates that we use to try to improve the gene correction using the repair templates. Another aspect that we are working on is on the gene edition of T-cell progenitors, because a lot of immune disease that we focus on are involving the T-cell development, and what we have set up is a platform to try to generate and modify T-cell progenitors that are differentiated from hematopoietic stem cells that are purified with a positive cell selection, that we differentiate on organoid, artificial thymic organoid, and that we are able to modify with CRISPR or prime editing strategies and reinfuse to the patient. So this is a theory, and right now what we are doing is to perform this kind of approach in the BLT mouse model. So it's an NSG mice that is engrafted with a human thymus. This is something that we are able to do here at Saint-Just with a collaboration with Eliadad Labs. We is mastering this kind of mouse model. So what we do is to isolate the hematopoietic stem cells, cultivate them on the artificial thymic organoid during 7 days, and perform the gene modification with the nucleoproteinic form of the Cas9 and the repair templates, and transplant the cells after 7 to 10 days of modification. So here an example of what we have done with for collaboration with Eric Cohen at the IRIX, who is working on the HIV infection. The plan was to target the CCR5 locus, to integrate in the CCR5 locus a CAR antigen driven by a SFFB promoter, and we've got different constructs here. You see we got a plasmid that allows the integration of any desired transgene, and right now we have set up this experiment to make the integration of a GFP transgene, and you can see that after 10 days of differentiation, we are able to do the integration of 20% of T-cell progenitor with our GFP transgene. And what we do is to engraft this T-cell progenitor in the NSG mice. The engraftment of the T-cell progenitor is enables the engraftment and the rapid generation of T-cell in the NSG mice, and a more rapid generation of T-cell than the control mice, and we are able to detect T-cells that express the GFP transgene at a certain significant amount of T-cell progeny in the engrafted mice. So that's basically what we are doing right now in the lab. We're doing that through our genetic platforms and our collaboration with, I told you, the lab of Eliadad, who is providing us with BLT mice, Dr. Eric Cohen, who is an active collaborator for this project with the CAR anti-HIV, and Christian Beauséjour lab, who is driving the iPSC platform when we have to perform experiment with iPSC cell lines. Thanks a lot.
Thank you, Fabien. Just a note, we are trying to open right now here in Padua the CDX1 trial you were mentioning at the beginning. So we are going back to your original contribution. So I just asked Professor Cristina if she want to share some of her activity, because at the beginning she just gave an introduction about herself, but I think she has a few slides. So Cristina, feel free to go. We cannot hear you. You're on mute.
Yeah, I'm trying to okay. I hope that you are able to see.
Yes.
Okay. This is just to introduce the topic. Are you seeing the slides?
Yes,
Yeah.
we can see them.
Okay. Okay. So this is the field where I'm high involved in the group, translational group, including genetician and biologist in the basic science setting, and cardiology, of course. Discussing with you the problem of inherited cardiomyopathies, which include both the traditional cardiomyopathies like hypertrophic cardiomyopathy, DCM, dilated or arrhythmogenic, and then the cardiomyopathies which are not structurally abnormal but functionally abnormal in terms of electrical stability, like channel disease. This is why, because these are diseases at risk of sudden death and ventricular arrhythmias. If we are now focusing the attention on inherited cardiomyopathies, you exclude a lot because, for instance, hypertrophic cardiomyopathy is not considered a rare disease. The prevalence is 1 out of 500. The same is for dilated cardiomyopathy, whereas arrhythmogenic is considered a rare cardiomyopathy because the prevalence is estimated about 1 out of 2,000. And the same is for ion channel disease, where you can have rare channelopathies, particularly some receptor variants. But back to the topic of today, which is the problem of, I don't know why I'm not able to proceed with the presentation. Let's see. Okay, you should probably see the slides now. They're moving? Yes. So the problem of gene therapy, we are far away from gene therapy in this setting. There are now target therapy because we are starting to understand which are the physiopathological pathway, the pathogenetic pathway, having assessed the genetic background of this disease, but with a few exceptions, we are really far away from gene therapy. The only example where there was some gene therapy for hereditary cardiovascular disease, you see the famous Marfan syndrome, the familial hypercholesterolemia, the primary pulmonary hypertension, paper in the literature, there are some clinical applications, and then focusing attention on my field, which is the one of inherited arrhythmic disorders, this famous publication from the Pavia group about catecholaminergic polymorphic ventricular tachycardia. About years ago, they developed a way to have a mouse model of catecholaminergic polymorphic ventricular tachycardia to modify with gene transfer the disease and the arrhythmic phenotype. So they were able really to reduce dramatically the arrhythmic presentation. And the good news is that the recent advances in gene therapy have been recognized, so that the US Food Administration approved the first gene therapy for the recessive variant of catecholaminergic polymorphic ventricular tachycardia. And these are some slides of the recent article from the group of Pavia. But now moving to the field, which is my field, arrhythmogenic cardiomyopathy. In Padua, we did a lot of progress to understand the genetic basis. We are now trying to understand the physiopathological pathway in animals. So we developed a zebrafish model of desmoplakin. And then we were able also to rescue with the pharmacological treatment the pathways. We studied together in collaboration with the John Hopkins group the desmoglein model of mouse model of arrhythmogenic cardiomyopathy, which is a model of biventricular disease. And we, together with the group of Marco Mongillo and Tania Zaglia in the biomedical department, biomedical science department, we are trying to develop now a mouse model of desmoplakin mutation of key mouse model. So work in progress, but there is not yet a gene therapy. Probably the most depressive part is that probably the genetic defect is not a delay only of the cardiac myocytes, but could be a multicellular level. So the recent understanding is that, for instance, arrhythmogenic cardiomyopathy should not be considered anymore just a cardiac myocyte disease, but probably also pluripotent stem cell, interstitial stem cell, monocytes, circulating monocytes are involved. So you're not just trying to correct the genetic defect at the level of the cardiomyocytes, but it should be a multicellular level. This is another, of course, challenge for gene therapy. So just to show you what is going on in the literature, you see here a recent publication demonstrating which are the existing emerging therapies for genetic cardiomyopathy. We know a lot nowadays about the genetic variants. The news that many monogenic disorders are not considered anymore monogenic nowadays, because we know the possible involvement of additional gene variants, SNPs, which can play a role in the phenotype of this disease. So there are primary effects in terms of loss of function. We can work here with gene therapies in terms of replacement, silencing, or somatic gene editing. But another possibility is to modulate primary disease pathways. This is For instance, what is doing they are doing now in the setting of hypertrophic cardiomyopathy, myosin modulator, protein stabilizer, think about amyloidosis, anti-aggregation agents, the same, or modulator of protein quality and control. And finally, there is the possibility to modulate secondary remodeling. So not just to correct the genetic defect, but also to act at the level of primary or secondary effect, prevent the secondary remodeling at the level of the heart. This is the example of amyloidosis. And as far as arrhythmogenic, this is the disease we are quite involved since many, many years in Padua because of the first pathological description, the first genetic description, the first family familiarity description in the cardiology setting. A disease which is genetically determined, rare, characterized by aggressive disappearance of the myocardium, the cardiomyocytes. Where is the defect? Only at the level of the cardiomyocytes, also at the level of the fibroblasts, adipocytes, and circulating cells. This is really still a question mark to understand the disease pathogenesis. We know a lot about the genetic background. So this gene has been identified in the genetic lab in the biology department or in our department. This is the first animal model, was overexpression, demonstrating the recapitulation of the human disease with progressive loss of the cardiomyocytes, inflammation, necrosis, and then replacement by fibrous and fatty tissue. The disease, although genetically determined, was not present at birth, but developed weeks or months later due to a progressive penetrance, disease-related, age-related penetrance. So at the beginning, you can have a normal myocardium, then you have necrosis with inflammation. So another target could be inflammation, immodulation. Then you have a fibrous tissue replacement. So another target for that could be the fibrosis pathway with TGF-beta, SMAD, and so on. And finally, you have the presence and the occurrence of aneurysm. The other model is the one I mentioned before, the desmoglein 2 model, the John Hopkins, where the investigation demonstrated a central role of this pathway in the pathogenesis of arrhythmogenic cardiomyopathy, and this is now a pharmacological target for therapy, but unfortunately still just in the lab, not in the clinics. There is no translation yet with this discovery. This is the paper I mentioned to you before about our zebrafish model of desmoplaking model of cardiomyopathy, which is rescuable by genetic and pharmacological intervention. And we should mention the important collaboration with the group Natasha Tiso and Francesco Argenton in the biology department. And finally, the one I mentioned to you before, recent study arrhythmogenic cardiomyopathy demonstrating that is not just a cardiac myocytes disease because mesenchymal stromal cells are involved in the genesis of the disease. Probably mutation are also, gene variants are also affecting this cell. And finally, the issue of a multicellular disease affecting cardiac and bone marrow mesenchymal stromal cells, again, in a model of arrhythmogenic cardiomyopathy linked to desmoglein 2 mutations. We are far away from gene therapy. Sure, we are still investigating the pathogenetic pathway, but some therapeutic targets are emerging in the lab, so that probably we can now work on this setting to stop not the onset of the disease, but at least the progression with patients at a younger age. Thank you for your attention.
Thank you very much, Cristina. So thank you all actually for introducing yourself and your research. I think we are representing a good proportion of disorders of potential interest for advanced therapies and for gene therapies, spanning from metabolic, where me, Alexei, and Leo are particularly involved, then immune disorders with Fabienne and cardiomyopathies. And I think all this testifies probably that the interest for advanced therapies and for gene therapy could be quite broad and could be shared by all of us in our respective disciplines. So the first question I've listed, I think we could already comment on, is the fact that possibly advanced therapies and gene therapy could be a unique opportunity in the rare disease space. And I'd like from each of you a comment on this and how you see this opportunity. Consider that for every of our fields, we might have to address the complexity of diseases that have complex pathogens and multiple target cells, multiple pathways to be considered when approaching them with advanced therapies. So I think we may start commenting on this with Alexei, if you agree.
Yes, sure. Yeah, that's really a difficult issue because one thing we have to deal with is the multisystemic involvement in this neurological MPS, because you have, of course, the major manifestations. The patients are dying from the brain disease, but actually you have issues like involvement of visceromegaly, you have joint problems, you have, for some disorders, it's the cardiac problems, and of course you need to target all these organs and tissues. Another very important issue is how you assess efficacy, because I mean, of course, we can all do preclinical work, but when you start thinking about the clinical translation, then you need to to see how you would actually convince the regulators that your approach is working, right? And for most, of course, you can monitor the for metabolic disorders, you can monitor the accumulation of metabolites, right? In case of MPS, that's different glucosamine-glycans, but these are not considered as the signs of clinical improvement. So you need to show that actually your disorder, your therapy delays progression of the disease or reverses. And this is very difficult because these disorders are rare or ultra rare. So you need to have a clinical history, and also it can be very different for different patients. So the approach is now we're trying to see if is to compare a patient to itself, right? So himself or herself. So like to monitor the progression like for a year before treatment, and then initiate treatment, and then compare for another. So whether this approach can be used for all disorders, it's hard to see now. And yeah, so, and then of course we don't know to what extent we can reverse pathology and whether we can reverse it at all. So my understanding is that for the aryl sulfatase A deficient patients, like those that were treated when they started presenting symptoms, they had prognosis, right, as compared to the presymptomatic patients. So that's another important issue to identify these patients as early as possible, and then kind of enroll them for treatment. Well, that's things I had to say.
Yeah, you touched many points, and I think we can go through all together most of them. To answer your specific question, yes, in the case of patients with metachromatic leukodystrophy, we can absolutely confirm that the factor that mostly affected outcome has been the disease stage at time of treatment. So at the moment, indication for receiving the the drug, say the registered drug, is just for pre-symptomatic or very early symptomatic, the latter only within the cohort of the juvenile onset, exactly because reversing neurological damage, particularly in conditions such as metabolic diseases, where we are just providing enzyme, we are not replacing dead neurons, is really not feasible. So timing, patient selection, these are criteria that came out to be key and critical for achieving benefit in the case of metachromatic leukodystrophy, and I will assume that it would be the same for other neurometabolic diseases. But as I said, this is just one of the many aspects you touched. And I would start from the basics. So if we all agree that advanced therapies and gene therapy could be a unique opportunity for rare diseases, we first have to agree about which are the criteria to select a disease indication for the clinical development and possibly future clinical development. And so I would like to hear, for example, from Leonardo, what you think about this. Which are the criteria we could use to select an indication as to be particularly promising for developing an advanced therapy, a gene therapy approach, and then we can discuss about modeling, in particular which animal models, which approach to select vivo, ex vivo. We have so many choices. How do we choose? What do you think?
Non si sente? You're not hearing.
No, I forgot to turn on the mic. Thank you for the easy question, Professor G. And this is the kind of questions that that would disappoint more the audience, because if you think of the patients group, everybody would like his disease to be the object of major gene therapy trial. So I think you have to consider several aspects. First of all, the frequency of the disease in the population, because certainly there's much more impact in treating sickle cell anemia than in treating coenzyme Q deficiency. I'm talking about my disease because we have few dozens of patients in the world, while there are millions of patients with sickle cell anemia. So that is, of course, one impact. Then I think is one of the other issues is the efficacy. So how really this treatment can modify and improve the quality. I don't like the term quality of life, but the clinical history of the patient. Considering that, is it really worth to use a gene therapy protocol, or we can use something else? Considering also that these therapies are very expensive. So I think that one of the issues is that these are treatments that should be offered if they're really beneficial. If they're, so, yeah, there is an improvement, and then I would be very, very skeptical about it. And certainly, this does not depend on the disease, but we have the experience of spinal muscular atrophy in front of us. We know that there are some patients who will really not benefit really from gene therapy, because the diagnosis is too delayed. We had a patient 2 weeks ago, and he was a 5-month-old patient with SMA1. The diagnosis was delayed, not on our side. Once it was not our fault. It took us 26 hours to provide a diagnosis, but when she was seen by the neurologist, she had very severe denervation. So treating a patient, even though the now spinal treatment for spinal muscular atrophy is an approved therapy, I don't know what the final decision was, but everybody was very skeptical in offering such a treatment to patients with so severe symptomatology. So these are the possibly some of the criteria. Then it depends on how powerful your patient association is. There are some patient associations that are very powerful, so they will push the therapy for their disorders. There are others that are less powerful, and they will have less impact. But again, it's very difficult to draw a line. But certainly, one of my point is the actual clinical benefit that.
I'd like to hear what Fabien thinks about.
Thank you for the easy question also.
I will answer myself.
I will bring my view from the immunology field, because we have plenty of history with the gene therapy, and it's very interesting, in fact, when you look back at what has been done in the field, because the first trials were really focused on very severe disease, like SCID X1 or ADA SCID, with an history of death for the patient if you don't do the treatment. So we had some alternative since the patient was transplanted, but the first thing and motivation to develop gene therapy in this field was to bring another treatment that will enhance the survival of the patient who had no suitable match sibling donor or match related donor. But here we are in after treating the patient for SCID X1 or SCID ADA. In fact, when you look at the other gene therapy trial, there was the trial with for treating the viscoeldridge syndrome, which has shown some benefit, but it's not so obvious when you look at the result of the gene therapy trial as compared to hematopoietic stem cell transplantation. And since in our field, gene therapy is always compared to hematopoietic stem cell transplantation, we've seen a lot of improvements in the field of hematopoietic stem cell transplantation. And when we compare right now some results of gene therapy trials to hematopoietic stem cell transplantation, we don't see really the obvious benefits of such approach. When you look at CGD, by example, it's a very nice example. You see that hematopoietic stem cell transplantation is able to cure the patient with something like 95% of event-free survival. You've got some people that will have a graft rejection, but you can make another transplantation with them and cure them with a good limitation in terms of adverse event related to the therapy. And you've got on the other end people treated with gene therapy trials for who there's some graft failure of the GTP, and you've got some people that will have some transient correction with lots of GTP, and others that will have just some kind of we'll say mixed chimerism, but partial correction that is probably not enough to prevent them to do infection. So there's a lot of discussion on the right focus to have and which kind of disease should be targeted. And on the other hand, I will stop with this and end with this one. You've got two gene therapy trials that are on the work of progression for the X-linked agammaglobulinemia, the Breton disease, which has been recognized in the early days as something like could be treated by immunoglobulin replacement. And now we got two gene therapy trial targeting this patient because we know that right now with looking back at the history of the disease, you've got a risk of infection, but also immune complication, auto-inflammatory disorder from the brain, from the gut, or the joint, and that you should have a benefit to do another treatment beside immunoglobulin replacement. But people won't consider hematopoietic stem cell transplantation for this patient, but they will possibly consider doing gene therapy because they think that it could help cure the disease. So there's, I think, a moving barrier and cursor in what kind of disease we can treat and we should aim to treat. It's a more difficult question than probably 25, 30 years ago.
Thank you. And Christina, what about the pathogenesis? Do you think that is because you explain us how complex could be the pathogenesis of some arrhythmic diseases, some myopathies where you have possibly many cell types involved. So how could this be somehow with the indication of developing an advanced therapy, a gene therapy treatment? Do you think, is there any space, is there any opportunity? And in case you do see that opportunity, what do you think would be needed? Advanced modeling, what?
Yeah, I mean, the effort that are put on to understand the pathogenesis of inherited disease in general is also because probably you can target not just the gene, but all the cascade of events. For instance, in the disease I mentioned, arrhythmogenic, there is evidence that there is a lot of inflammation at the beginning and also during the evolutive phase of the disease. So there is now a lot of research, basic science, to understand which is the trigger of this inflammation, whether there is an immune reaction against some antigens. So, okay, you cannot correct the gene defect, but you can address probably these phenomena, so you can delay or stop the progression of the disease. And there is evidence also the mouse model. Okay, mouse model is not human disease. You know that they are not fully representative of our disease. This is, of course, a problem. But there is some early evidence that if you treat this patient with immunosuppressive therapy, you can delay the disease progression. So this is another opportunity, just not to say you can just make a somatic therapy with the antiarrhythmic drugs, with diuretics, with inotropic drugs when there is a cardiac remodeling, so preventing cardiac remodeling. Of course, our target, final target, is to prevent the disease onset. But the problem is also when we work, particularly in Italy, at the germline, at the origin of the disease. You understand what I mean? Are we ready to do this? There were some publications in hypertrophic cardiomyopathy about this, but then still discussed, of course, this topic. Probably we use an ethical discussion on this.
Thank you. And what about you, Maurizio? What do you think are the factors that are playing a key role in selecting indications for the development of advanced therapies and gene therapy? We cannot hear you.
Can you hear me now?
Yes.
Well, as a matter of fact, if you allow me, I think it would be interesting to share with you my experience regarding the evaluation of efficacy, because I had the opportunity some years ago to participate in the evaluation of Glybira at the European Medicines Agency. As you know, this drug was developed for the treatment of lipoprotein lipase deficiency, and it became the first gene therapy drug approved in the market. The clinical study By the way, the clinical study was performed in Canada, as far as I remember. It is a rare disease affecting about one patient in 1 million in the population. And these patients are on a very strict diet since they develop pancreatitis. If they don't adhere to the diet, they develop pancreatitis. And the evaluation of efficacy was based on the incidence of pancreatitis. Each patient was the control of himself or as self, as Alexei mentioned. But the issue with the commission was that the statistical significance couldn't be reached. And well, there were some attempts to reach this significance, but there was a discussion going on for 2 years because the statistics were not convincing. I personally spent several days trying to going through the clinical dossier, and then I realized that these patients, all these patients following treatment, were gaining weight. Yes, they were developing also to some extent pancreatitis, but they were gaining weight, 10, 12 kilograms. And in a patient with lipoprotein lipase deficiency, this is absolutely unusual because as soon as they eat a little bit more, they feel bad, they develop pancreatitis. So this was an indirect evidence that the gene therapy was working because the tolerance of these patients to diet had increased. So the target was not correct. This shows that when we are approaching this kind of clinics, our view needs to be less rigid. We need to show more elasticity, and as a matter of fact, it was because of this observation that finally the Commission approved the drug for the market. I think it can be of interest to share this personal experience because it's really, I think sometimes we need to change our paradigm that tend to be too rigid in the evaluation of these diseases and of the treatment, of course.
Thank you.
Yeah. It's very interesting. May I ask at what age most of patients were treated? So was it like gaining weight more like a normal development, or they were?
No. These were all adult patients.
Oh, adult, right.
Yes. And what is interesting is that before treatment, their weight was stable. At least they were followed for an average of 18 months before treatment, and the weight was stable. But after treatment, most of the patients were really gaining weight significantly, as I said, up to 10, 12 kilograms, which is a lot for this kind of patients.
Thank you, Maurizio. I think with this conversation, we can really go back to what Leo was commenting at the beginning, and I leave you the discussion, but I think you want to comment about the likelihood of benefit and the perception of benefit, right? But we cannot hear you.
Sorry. What I'm saying is that it's very provocative in some way, but I had an old professor who used to say that if you need statistics to prove that a drug is working, it means that it's not working. And I mean, this is, I mean, nobody had to do statistics on penicillin. And this is, I mean, I know it's, I'm just being provocative. But one of the issues here is we are dealing with extremely expensive treatments, even because when they come out of the mental stage, they are extremely expensive, because one cycle of therapy for SMA is €1 million. Correct me if I'm wrong, Alessandra. So, I mean, this treatment must be really doing something clearly positive for the patient. I think that is the point. Then the way to demonstrate is what correctly Maurizio was pointing out. But I think that it needs to be something like water of Lourdes, something that has some miraculous effect in order to be something that we should apply to the treatment of patients. Otherwise, it will never justify the incredible cost that it's for the community. And in a time of limited resources, I don't know how we'll deal when more than 20, 50 therapies are apparently about to be approved. How can we provide? The only reason why that we should offer them to the patient is this really its efficacy is really proven without any doubt. I don't know if you're right, if you agree, Alessandro.
I mean, there are many aspects, and some of the points you raise could be applied in different stages of treatment development. And at each stage, they could be perceived differently. So if I can try to go through what you said before and also now. So we were discussing which could be the factors that could lead to the investment or to the choice of specific indication as a candidate for development of an advanced therapy of gene therapy. This choice, you do it up front. And you do this choice based on a predicted or expected benefit that obviously has to be proven, first of all, in preclinical trials and then eventually in the patients, based on results of a similar approach in similar diseases, or on your ability to somehow anticipate, based on the mechanism of action of your therapy, potential outcome. But that's just predicted. The other factor you mentioned is strength of the patient's community, of the patient's associations. Well, this component can have a different perspective of efficacy as compared to your or my perspective, or the perspective that someone working not in the space of gene therapy could have. So, for example, if you tell me that it is worth to develop this kind of treatment only for diseases for which the outcome is supposed to be really changing, dramatic, then a patient association may not agree with this, because a strong patient association could support the development of a treatment for a disease in which you would expect just limited benefit or minor benefit, like extending the life rather than maintaining the quality of life of the patients. And so it's difficult to reconcile the portion, the investment, maybe the funds that the patient could provide for you to develop gene therapy for that indication, with your expectation that, for example, in that indication, the treatment might not be life-changing, it just be prolonging life. So these factors are very difficult, and it will depend on who's involved in the discussion and the stage of the discussion, because, for example, you mentioned frequency. Frequency could be a factor if you are talking to an industrial sponsor that could be interested, particularly nowadays, is just interested in gene therapy for diseases which have a broad market, because after CAR T cells, which told us that gene therapies could be for a large public. Nowadays, all the biotechs in the field just look for large indications. They no longer care about NLD, Lipo, SCIDEX1. For us, for an academic stage or for the patient's association, frequency is not one of the factors. So all these factors have a different weight according to the stage you are approaching. If you are just at the beginning, if you are thinking or not to develop your robust proof of concept of clinical study into a clinical application, or whether, for example, you have completed a phase 1 clinical trial and you need to move forward. So it's very difficult. It depends on the setting. It depends on the context. For sure, what I can tell you is that really in the process of moving from the preclinical to the clinical, it could be very difficult to predict the extent of benefit your treatment could provide. And I just tell you this story, which is very personal. When we developed the first application of our HSC gene therapy to neurometabolic diseases, we had a very weak animal model of the disease we were interested in. It basically reproduced just a little bit of disease, minor, normal lifespan, no behavioral deficits or no obvious behavioral deficits, weak histopathology. We obtain robust evidence of benefit by our gene therapy in the proof of concept studies. Some people in the field, they said this is not meaningful because the model is weak. This would never work in patients. Anyhow, we were convinced it could have worked in patients, and so we built it our clinical trial. And when we designed the primary endpoint of efficacy, we designed it as very weak because we were worried and we thought we would have seen minor benefit. We could have probably just prolonged a little bit the life of the patients, but we were very scared we would not have seen real life-changing effects. In reality, that treatment, which now is commercialized, resulted in dramatic benefit. I just received yesterday the picture from the 6th patient we treated. She is now 11 years old. She was treated when she was 16 months old, and she's going to school. She's having a great life, normal life. She has the same age of my son, and they're going to school both. She's doing sports. She is having a normal life, and her sister with the same disease, she died when she was 6. So we would have never predicted that. So it's really difficult. And nowadays, with that experience and other similar experiences, we might say that in lysosomal diseases with CNS involvement, HSC gene therapy could possibly work at user strength, but it could vary in reality from one disorder to the other. So it's actually very difficult. So if I had to answer to my own question, I would probably say that I would choose an indication based on a strong pathogenesis, so having very clear in mind what the pathogenesis of the disease, and knowing that the approach we are undertaking, the gene we have selected, the cell type we have selected for gene correction, indeed impact on that pathogenesis. That could at least let me think that we have an expectation for impacting on the disease and so for predicting some benefit. And probably I would also look at potential patient access and potential marketing development, because what we have learned over the years is that what we are doing to be meaningful needs to lead to a drug. And to lead to a drug needs to find a commercial partner and the opportunity for marketing development. Fabienne was referring to the first trials of gene therapy in SCIDEX1, so severe combined immunodeficiency due to gamma chain deficiency, and ADA SCID. Well, for SCIDEX1, we still don't have a drug. We have a clinical trial, the one Fabienne was referring, we are trying to open in Padua, which is still academic, which is funded by the NIH in US, and they are opening right now multiple sites to enable patients enrollment, which is open at Dosh. We are trying to open it at Padua, but it's totally academic. ADA's kid, we had a drug on retroviral vector, which is no longer on the market for severe adverse events. 60 patients have been treated with lentiviral vectors, but no drug as well because it didn't progress after marketing. So it is very complicated. It's definitely very complicated, and at least having the perception that what we are doing, the disease we are selecting, the approach we are proposing, could have some chances for development up to registration is a criteria because it not only confers some feasibility, provides some expectation of feasibility of moving forward, but also some meaningful context for what we would be doing. So it's definitely very complex. It could be complex to decide what approach to use. So we have been discussing about, I think Alexei was referring to integrating vectors, lentiviral vectors very likely. Then Leo, you were talking about AAVs, adeno-associated vectors, and Fabienne was talking about gene editing. How can we choose? Who wants to comment? Alexei, do you want to say something about it?
Well, yeah, I actually wanted more like to follow up on what you said. I think your story is very inspiring, and in a way, I think it worked perfectly. It was first, and it worked very well. And it reminds me like when the first enzyme replacement therapy for lysosomal disorder, it was developed for Gaucher, where you have mannose receptor, and the most affected cell is macrophage. So if it would be another disorder, probably it wouldn't work that well, and then it wouldn't inspire all the other developers to follow up. So that's really was in a way that was very lucky that aryl sulfatase deficiency A was selected. But for the choice, yeah, I wanted to share my experience that a lot of companies now are jumping on the AAV9 vegan. So like basically even developing treatments for disorders that are like non-neurological, Hunter disease, or so you see when now you visit like at the Congress I attended last week, you see multiple exhibitors showing that we also have AAV-based platform, and we want to do the treatment for a Fabry or a Hunter or a Pompe, and so I think in a way that I think it's like AAV should be really carefully considered before people start using it, because we see, for example, there's a AAV immunity, right, to the capsid, which naturally exists in the majority of humans, right? So that should be also considered before selecting this. Then, like I think the downside of either ex vivo or in vivo therapy is that we cannot carefully dose the transgene, right? So in a way, in lysosomal field, we're lucky because we don't see negative consequences of overexpressing, of overdosing the normal lysosomal enzymes. So maybe they are, but so far we didn't see them. But
some that are absolutely
yeah. But for the other disorders, it may become an issue, right? If you overexpress and then you can severely shift the regulation. So in this way, the repairing the mutation, it looks safer, right? Because we're kind of restoring something that existed before. However, this so far it is limited by the low efficacy of CRISPR approach, and also like existence of off-target defects. So there is a balance, right? And of course, I mean, like there are different new approaches, right? That for gene correction. But again, when you increase efficacy, you also increase the probability of off-target effect. So I would say, and finally, there's an approach when you try to engineer, like you try to insert into the albumin gene the enzyme that you need, so to convert like a liver to a small factory for producing this. But so far, we didn't see any clinical proof of efficacy for such approaches. So I think to choose, it should be you should start from the disorder, right? And also whether you need to correct only several cells, or whether you need to correct most of the cells, and whether potential pluses and minuses of doing the bulk gene correction. Thanks.
Thank you. Fabienne, what do you think? You move into gene editing, right? That's your area of interest at the moment.
Yeah. In fact, I still think that there are a lot of room for continuing approaches with viral vectors correction. The gene editing is interesting mainly, I think, for genes that need a proper regulation. We made, even if we made a lot of improvement, in modulating the expression of the transgene with viral vectors, because you can still design and use some specific sequences or an answer to modulate the expression of the gene. You still got disease where the context of the genomic context of the gene is important, and you need to have on-site correction. And it's especially true with gene affecting signaling pathway in immunity. If you think to STAT defect or JAK defects, probably the only thing that you can do is to target those genes with gene editing stuff. So the problem is that with gene editing is that the level of correction still remains low in view of the desire of enough correction in the targeted cells. And we, despite improving, the efficiency, we still are far below the objective of complete or near complete correction of the cells that we need to correct to have an effect. So it's still complicated. And as discussed with Alexei, there's still this problem of targets. Okay, even when you design proper guides or you try to lower these things, there's still this worrying effect of gene correction. And for what I see in a recent clinical trial, it's still not completely, I see, sufficiently addressed, in fact, in my mind. I don't see as good, I would say, investigation of follow-up of off-target effects in gene therapy products modified by CRISPR, as we have seen in terms of integration site analysis for the gene correction for all the SCID trials, by example. You see of target analysis on the gene therapy product, but you don't look at potential off target in the progeny during the follow-up of the CRISPR study. So it's kind of a worrying things for me.
I think there are many issues in both sides of integrating vectors versus gene editing, both in terms of efficacy and safety. And again, specific indications may require dedicated choices. So I think it's very difficult to just decide whether to work on a specific vector or on editing per se, because everything depending on the setting, on the target gene, on the target population may have specific requirements. Given the fact that whenever we cut the genome, either by an integrase from a lentivector or by CRISPR, we run in some risks. And the risk would be proportionate also to the characteristics of the cells we manipulate. So whenever we have a sort of predisposing condition to transformation, or wherever we have a specific targeting by our gene therapy approach, we may be there's a risk that all of these approaches share.
Well,
you know, Christina, that AAVs, there are some serotypes of AAVs that are very, very good at transducing cardiomyocytes. Do you have any clues on applications? I don't remember the indication actually. When I was at BCH, at Boston Children's, there was a group of Dr. Hu, which was actually developing a gene therapy approach for an inherited arrhythmopathy using AAV. And it was very interesting because we were working on a potential translation of these clinical findings in mice.
You're referring to adenovirals.
Yeah, associated viruses, which are derived from the adenodes. They are much smaller, and there are many serotypes that can interact with different cell types. And there are serotypes that are very good at transducing cardiomyocytes. And there are some nice experiments, at least in mice. I think there's something in rabbits showing some impact on inherited cardiomyopathies.
Leonardo.
Well, if I may add a last comment, I think besides the technicalities, there's a main difference in technicalities. I mean the problems of the off-targets and whatever between gene replacement and gene editing. And the issue here is basically that if you develop a gene replacement protocol that is for, in case of a recessive disorder, that is good for all patients, while the gene editing protocols are mutation specific, and that is the main problem.
It depends. I think what
you can sort of adapt them, but or you can adapt them to knock out specific alleles in case of dominant negative disorders. But if you develop a treatment of for example, for cystic fibrosis, you can try and correct the F508del mutation, which is in 50 to 75% of the alleles in depending on the mutation. But the patients who don't have the F508del will not benefit from your
I will say it depends on the approach of gene editing, because you can still envision to insert the corrected cDNA in front of the promoters. So it could be a multi.
The efficiency is even lower, and we do it in yeast because rather than doing it now, we do all our yeast clones using CRISPR-Cas9.
I completely agree. The problem here with this method is completely the level of efficiency of the gene correction.
Yeah, but again, it's a technical problem. I mean, we are doing this kind of experiments on the human hematopoietic stem cells of targeted gene addition at a locus we like or at a safe locus like AVS1. And you reach efficiency that in vitro, on the in vitro progeny is around 50%, 60% if you're lucky, and if your gene editing expert is very good. And in vivo, you can keep seeing 30, 40%. So what we see in experiments and what we see in the let's see, in a setting that could be predicting what would happen in patients is that even targeted gene addition could be working with some effort, with some large manufacturing improvement. I think the point is more on a strategic point of view, both for in vivo, ex vivo approaches. Strategically, which are the cells we need to target? Which levels do we require? Do we need regulation of gene expression? These are the factors we may use to decide. Unfortunately, not safety, because more or less, as I was mentioning before, we do expect some effects because we are cutting the genome, except for AAVs, which remain episomal for the majority. But then we have the other issue, AAVs, and maybe there you want to discuss about this, which is the fact that, particularly if you administer them to young children, most of the cases they get diluted over time, right? Leonardo, do you want to say something about this?
Well, I don't know. I'm not such into in the topic to provide really insightful. And so the only thing that I think is that ex vivo therapy at this point seems the most promising, is probably the most promising, because it's the ones that reduce toxicity, and with the new vectors that prevent integration in critical sites. This is probably the closest. The approaches, these kind of approaches are the closest to be implemented in clinical practice, whereas systemic injection, especially in adults where you need huge viral loads, are the ones with the approaches with the toxicity. Apart from that, I am not such an expert. I mean, so I don't want to. I rather stay silent and let you think that I'm ignorant, and then speak and then prove that I am.
So why did you choose AAVs for your application?
Because those were these are basically the only virus that can transduce the kidney. The lentiviruses won't work, so it was the only solution available. Then in our model, to have also transductions of other extrarenal tissue is not a bad idea. These patients have also partial deficiency in other organs. So the issue is that the kidney is the one that responds best to supplementation. So the reason why we got into this project, and I have to say, because I didn't say it, that is prednisone is specifically following this issue. The reason why we chose this, because it was the only of our disorders that could be approachable by such techniques. The other mitochondrial disorders are extremely severe, and so once you diagnose a patient, it's probably really very late to do something. We learn it with coenzyme Q deficiency. On the other hand, the other metabolic diseases are more popular, so there are already many people working on them. So it wasn't a good idea. I know nobody working on coenzyme Q deficiency. That is why we chose this approach. And again, the reason, if anybody has any suggestions, I am here to take them on how to improve or whether to change because the literature is quite scarce, and there is this really very few papers. Actually, with the nowadays that we can have fast diagnosis of renal disorders, and gene therapy for these disorders could really be something that could change the natural history of these disorders. I've always been skeptical about gene therapy in the past years. Now I said I went back to Canada. No, I think that the technology now is mature enough to warrant some, even from people not vectorologists as we are, is try and play around with these issues. I think that in the past years the issue was to develop vectors. Now that there are better vectors around, you can try and see whether you can apply it to wider set of disorders. This is what I think,
so if this is true, and I strongly believe it's true, Maurizio, which are the challenges to go to the clinics?
Well, I don't think I have much to add to this interesting conversation, Alessandra, because, as you know, I'm not an expert in the field of gene therapy. As although I am having and I had some experience with the regulators and with the regulatory agencies. I think that I'm not saying anything original here. The first thing is to engage as soon as possible with the regulatory authorities, ask for scientific advice, make clear critical questions, and then be especially critical with as I tried to explain, with the clinical with the measurement of clinical efficacy, because in the experience I had with the European Medicines Agency, I saw that usually the problem rise there, because usually the methodology that is applied to demonstrate efficacy is not considered adequate enough by the authorities. I remember another case where the isotopes were used to demonstrate a metabolic effect, and again there was a methodological problem. Now I'm sorry, because I was not really prepared to answer such a complex question. This is this is a field I am not specifically working in, as you know, and my experience with the regulatory agency dates to some years ago.
But for sure, the regulatory part is a challenge and is a challenge that has to be addressed soon enough. Even in the late stages of your experiments in mice, because built in the proper way, some of the preclinical studies, in agreement with the regulatory authorities, somehow ensure a safer path to the clinics. What do you think, Fabienne? You have experience in this transition from clinical to clinical, right?
Yes, it's always a complicated stuff to transition, I will say. It's always complicated because it is not once you've got your idea and you've got your clinical data, the problem is the scale up and to get the funding for doing the scale up, that is quite expensive. So you've got to find a way to get financial support. And I see when I was back in Paris, we had some support from the Geneton of our institution. But here, when I'm looking at in Canada, it's way more difficult to get funding for this part, because you cannot get great funding from your regular funding agency, because you already prove your point by doing your preclinical stuff, but still you've got to spend money to do the translational part, and to get funding here is very complicated. So what we are trying to do right now at the level of the Quebec is to try to develop some kind of support from I will say private funding through foundation that will help us to develop some projects that we think are worthy to support to do this preclinical study, which is a kind of a GLP toward GMP scale up. And for this, you've got to have good preclinical and scientific data, and you've got to have also good partners in terms of gene therapy facility that will be able to produce your preclinical batch in a GMP compliant manner. So we are trying right now to figure out how to proceed clearly, because it's really the most difficult part once you've made the validation of your point in your preclinical data to transition to the clinical part. We've got a lot of discussion already with our regulatory agency, but the point is not really to mount the dossier or to write the IMPD. The point is to really to try to produce the preclinical batch and prove that you are able to do it for a clinical trial, and to get the funding for the clinical trial. So it's kind of a complicated situation for, I guess, every scientist that is involved, who is involved in this field. I think Alexei has got the same problem as we.
Well, yes, I think not only me, but yeah, unfortunately, in Canada, we're a bit behind in terms of GMP production or GLP production for the vectors. I mean, like a lot of facilities are talking about this, but like it's hard to find somebody who is certified. Second, yeah, the issue of to find funding is very important, and there are basically two ways to go, and one is investigator-initiated trial, which can actually be funded by research foundation by patient's foundation, by the government agency, or it can be privately funded, but this company, this industrial partner should be funder, not a sponsor of the trial. Then in this case, the trial is still controlled by the investigators. Another way is to have a partnership with a private fund company and have them as a sponsor, but which has negative downside that actually you lose some way control how this trial is organized. And yeah.
So manufacturing and financial resources. Can I ask Cristina your opinion? You are less in the gene therapy space, but maybe you can give us, let's say, a different view. What would you wish to see in order to consider sending a patient with an inherited arrhythmia to be enrolled into a gene therapy trial? What are the let's say, the information you would wish to receive in order to consider sending a patient into such a trial?
I don't know, because they are not perfect, so probably I missed some words. But so the question is exactly what I wish to see.
Yeah, what would be convincing for you in terms of the clinical, manufacturing, trial design, whatever, for allowing you to say, this patient of mine who has that specific cardiac disease, I'd like to send him to be enrolled in that gene therapy trial? Do you have specific information that you think would be critical for let you consider that as a real option for a patient?
It's a very difficult question, of course. The problem is that we see a lot of family members of people with severe arrhythmias or even sudden death episodes. So what they ask us is there a therapy to stop our disease evolution? So the issue is what we can advise to these patients or these family members at present, because when we say there is genetic therapy, we can modify, we should be very careful, because patients are really looking for a solution for them and for their family members. So these patients have already the disease. The problem is we are facing what I don't know if Leonardo, this definition is still used as carriers, because you healthy carriers. So you have a patient with a classical phenotype, maybe who died unfortunately due to arrhythmias, and then you genetic screening, you find a variant which is really pathogenic, and then you identify this variant in a patient, in a family, not a patient, just a family member carrying this mutation or this variant, sorry. So it's not yet a patient, is asking us something to prevent the disease onset. So really they ask what I can do, modify my lifestyle, or is there a drug that can stop the disease penetrance and manifestation despite genetic defect is there? So this is the real question they are asking us. As far as the disease, we are, of course, familiar with, because they are the classical one we are every day discussing with a cardiology, a genetician. We don't have yet the answer for this, apart from lifestyle modification to prevent this cardiac modeling, so to reduce effort, stress, because stress is, of course, making an overload to the heart and so on. But we don't have yet the answer for them. So this is why we are looking not just for gene editing, to target the gene defect, but also to work the pathway, the pathogenetic pathway, because this could be a solution in this carrier of the disease, the gene defect.
This is a key point. If I look at my field, which is metabolic diseases, sometimes you have the same situation, and I mean, patient has a mutation that is associated to the specific disease, but you cannot predict when the onset would happen, and what would be the severity of the condition. Tomorrow I'm seeing a patient with with Alberto Borrina that not very well for metabolism. This patient was identified by newborn screening as being affected by Krabbe disease, which is one of the most severe lysosomal diseases. But he has just a little bit of the substrate instead than a lot of substrate accumulated in his cells. This means that we don't know when the disease would manifest and how severe it would be. And so we don't know whether we should plan this to go forward immediately as we would do by routine, or whether we would wait. And the same choice applies to gene therapy. For example, the disease I was mentioning before, this MLD disease, we worked on by gene therapy. Most of the patients we have treated thus far and would be treated by this drug would be pre-symptomatic. It means children that we identify at the moment because they have an older SIB affected that we cannot help, so they have the mutations, but they have not yet the symptoms. So we treat them with the knowledge that at some point they would be sick, but they are not yet sick, and we expose them to the complications and type of the treatment. So this is very challenging. So knowing more about all the other factors that contribute to the phenotype is definitely relevant in order to really identify the patients who would benefit most from our advanced treatment. Particularly now that we have newborn screening being extended to so many diseases, and for some of these diseases, we don't want to tell to the parents. We don't know what the implications of a positive screen. So that's why when I was talking with Christina, we were saying that gene therapy and advanced diagnostic really have to progress together, because they really influence each other. Being able to offer complex and costly and costly treatments which are associated to side effects, as we are having very clear in mind the patient's population we propose them to, and to identify the right patients, we need diagnostics, we need genomics, right, Leonardo? You are definitely more than this, right?
Just wanted to ask what panel members think about perspective of in utero gene therapy, because we know now some enzyme replacement is done in utero, and in case that we had prenatal diagnostics and the case is very severe, so there isn't no another option. If so, like it's done through the vein, umbilical vein. So
Yeah.
what do you think?
Oh, so the first papers on in utero gene therapy, I think they came out in 2000 or so. So it's at least 20 years that people are talking about this. Most of the work was done in mice. There was some interest at that time, then the interest faded away. More recently, they are still they are talking again about this. For example, at Harvard, there is a strong program, a robust program on fetal medicine, and they are absolutely convinced that within fetal medicine, gene therapy should have a key role. I think it's very challenging, not only because in utero we know even less about our patient, because, for example, for metabolic diseases, our biomarkers, we don't have them, because the patient's blood is in equilibrium with the mother's blood, so the mother carries for enzymatic activity. The same is true for immune diseases or hematologic disease. And you have more barriers to gene transfer or to cell transplantation. The concept that the fetus is immunotolerant, I think, is still debated. So it's really complicated, and I think our technology still has to be handled. The patient outside of the uterus, I think, is still far away from being ready to handle an in utero gene transfer. At least that's my opinion.
If I may comment, our experience, because we provide prenatal diagnosis for most of these diseases, and even for those for which there is an approved therapy, and for which this patient will be treated immediately after birth, still, I didn't do many statistics, but the majority of the parents would choose for this termination of pregnancy. This is our personal experience, even providing them really neutral counseling and letting them speak with the people who would actually administer the treatment afterwards. So this is one point. The other comment I wanted to make is that for metabolic disease, we have done a lot of genotype phenotype correlations and tried to develop systems for genotype phenotype correlations. For certain patients for which we have the neonatal screening data that were identified for two neonatal screening, we were able to sort of predict, make quite accurate predictions. But there are some other genes, it's disease specific. It's clearly disease specific. For example, we have observed clear phenotype correlation for argeniosuccinic aciduria, whereas for citrullinemia, that's the previous enzyme in the urea cycle, we found no whatsoever genotype phenotype correlation. So it is really challenging. And when they talk to me about medicine, I laugh, because we still cannot understand why patients with the same mutation in ABCD1 have either natal disorder, or there was a patient that had a truncated mutation and was 72 years old, but from Paulina's brothers, she was sent it to me and said, look, he has a truncated mutation, can't be as he can be, so don't worry, because I thought we had converted the samples during the run, and but again, this is still one of the main points. We have really more. We're going to talk about artificial intelligence in genomics, but we have a lot of ignorance at this point in genomics, and this makes taking all the decisions that you were talking about much harder.
Thank you, Leo. I have a last question, and I would start from Maurizio. And the question is, what do you think about the role of networks and collaborative projects for the development of advanced therapies in rare diseases? And this would encompass also extracellular vesicles and in general, this area of therapeutics that frequently has the need of patients affected by rare diseases.
Well, I have always relied on networks and cooperations for the development personally of the projects I had the opportunity to participate. Networks are fundamental because in such complex disorders, usually you do not have all the tools that are required to successfully address the problems. So you really need to gather different expertise together, and sometimes it's a matter of how do you say? It's not black and white. It's a matter of shades sometimes. Slight differences in expertise can make an enormous difference in the resulting approach. So my answer is it's absolutely fundamental.
Thank you, Maurizio. Anyone else who would like to comment on this aspect? Have you been part of efforts within consortia that have led successfully to the development of either new indications or new therapeutics?
And by the way, sorry, we should never forget the importance of the collaboration with private companies that are essential, because without a private company with experience in manufacturing, it's extremely difficult for an academic group to reach the clinic. And often we realize this when it is too late. These cooperations have to start early enough.
Absolutely. Probably, if I could anticipate the concept, the large networks are fundamental, or could be fundamental, to give access to large populations, natural history data that could really be fundamental for developing a translational plan for confirming a disease indication, right?
Yeah, absolutely. I think it's crucial. I mean, like when you can rely on academic collaboration, but for more preclinical studies, and but for translational studies, I think it's crucial not to repeat somebody's mistakes, and just yeah, I think that in particular this workshop I hope will lead to certain collaborations. I was really interested to learn about this Maurizio's expertise in extracellular vesicles. We were not that much interested in them before, but now it looks like we are, and I hope I would be able to approach and approach you, Alexander, concerning the vectors and so on. Yeah, I think that's very important.
Great. So thank you all. We had a very fruitful and interesting discussion. If any one of you has anything else to discuss, we can, I think, close the session. That was always interesting and very valuable. Okay, great. So thank you, everybody. I think we are done, and I really hope to have the chance to meet you soon and to collaborate.
Thank you.
Right.
Thank you very much, everybody.
Bye. Thank you.
Thank you. Bye.
Bye.